In cardiac research on adult zebrafish, drug treatments are usually done in a systemic way. With intrathoracic injection, one can locally deliver solution to the heart without causing myocardial damage.
This method allows the delivery of small volumes of drugs, proteins, or other molecules in solution precisely around the heart of an adult zebrafish.
To begin this procedure, use a needle puller to pull microinjection adapted borosilicate glass capillaries. Store the pulled capillaries in a 9 cm petri dish with rails of modeling clay or adhesive tape. Using common scissors, cut a piece of sponge and carve a fish-like silhouette in its middle. Next, prepare small aliquots of the injection solutions with the tested proteins or other compounds. Adjust their concentration depending on the assay by diluting the substance in 1x Hangst balanced salt solution complemented with 10% phenol red. To obtain the working concentration of anesthetics, add 2 to 3 milliliters of tricane stock solution to a beaker containing 100 milliliters of fish water. First, turn on the stereo microscope with the light from the top and adjust the magnification to 16x. Soak the sponge with fish water and place it on a 9 cm petri dish on the microscope. Adjust the focus, and under the stereo microscope, use iridectomy scissors to cut the end of a microcapillary approximately 7 mm from the base, as detailed.